Of the ways of knowing in the theory of knowledge, the natural sciences rely most on reason. There are two types of reasoning, inductive and deductive. Inductive reasoning is the process by which specific observations are used to draw general conclusions, specific to general. Deductive reasoning is the process by which a general conclusion is used to predict specific outcomes, general to specific. Both get used in the natural sciences, but we will usually use deductive reasoning. What this means is that we will take a known theory and use it to explain why something happens. We can do this using natural selection to explain antibiotic resistance in bacteria. Before doing this, let us look at the ways in which we can stimulate our immune system to prevent the spread of infectious disease. One type of substance that could be used in preventing infections are vaccines. Vaccinations contain inactive pathogens, bacterial, viral, fungal, and parasitic, that are injected directly into the bloodstream. This stimulates the primary immune response and the development of memory cells. During this time, a patient will show symptoms of the infection, but not actually be infected. Also, the immune system is producing B cells that will fight that infection if infected by that pathogen. For example, you can get an influenza vaccination to prevent the flu. However, the flu vaccine only targets specific strains, so it will not be effective against all forms. Getting vaccinated yourself also protects people around you by limiting the spread of disease, especially to younger and older people or people with suppressed immune systems. A second common substance that could be used are antibiotics. The first modern antibiotic, penicillin, was discovered on accident by Sir Alexander Fleming. Antimicrobial substances were used centuries prior to the discovery of penicillin in 1928, which you can find out more from Radiolab's episode titled Staph Retreat. Antibiotics are substances or compounds that kill or inhibit the growth of bacteria by targeting the metabolic pathways of prokaryotes, but leaving eukaryotic cells and viruses unharmed. This is due to the effectiveness of antibiotics against prokaryotic features including key enzymes, 70S ribosomes, and the bacterial cell wall. Viruses lack metabolism and therefore cannot be treated with antibiotics. Some strains of bacteria have evolved with genes that confer resistance to antibiotics, and some strains have multiple resistance. Viruses do not carry out metabolic reactions themselves, but instead infect host cells and take over their cellular machinery. Instead, viruses need to be treated with special antiviral agents that target features specific to viruses, like reverse transcriptase and retroviruses. Since the advent of antibiotics, there has been a growing trend toward antibiotic resistance in bacterial populations. Currently, the World Health Organization recognizes 3 bacteria considered to be of critical priority, 6 considered to be of high priority, and 3 deemed to be of medium priority. How can the mechanism of natural selection for the theory of evolution be used, then, to understand the development of antibiotic resistance? First, there has to be variation in the population of bacteria, where some possess the gene for resistance, usually found in the small circular plasmids. Other bacteria in the population do not possess the variation granting antibiotic resistance. If resistance is not present in the population already, mutations in genes can create new alleles that produce antibiotic resistance. The larger the population, the greater chance exists for random mutations. Bacteria can also transfer genes between individuals, which can spread the proper allele through the population much faster. Through binary fission, these bacteria reproduce and pass on their genes. Each bacteria is reproducing, causing the population to effectively double in numbers after each division. This produces more offspring or daughter cells than what our bodies can support or fight off. The structure and function of our immune system is adapted for fighting off infections without the use of antibiotics, and this is when we begin showing symptoms. In response, we experience fever and chills, mucus production, coughing and sneezing, antibody production, and phagocytic macrophage attacks to destroy the bacteria causing infection. However, some bacterial infections cannot be effectively controlled this way and antibiotics are needed, like in the cases of syphilis or chronic strep throat and sinus infections. Now the antibiotic is taken, specifically designed to target the metabolism of bacteria causing the infection. This is a selective pressure as it affects the bacteria's ability to survive and reproduce. The antibiotics are highly effective against non-resistant members in the population and less effective against the resistant ones. As the population is reduced in numbers, our immune system is able to support the elimination of the infection. Symptoms start to disappear, although there may be members of the population in your body. These members, despite having a microscopic war waged against them, are still reproducing, including the antibiotic-resistant members. So, those with the genes granting resistance will be better suited to this change, be more likely to survive, and more likely to reproduce. Over time, the population of bacteria with the right alleles will increase. The greatest risk of spreading antibiotic resistance is the widespread use and misuse of these medications, such as overprescribing antibiotics when our immune system could have fought off the infection without medical support, misusing antibiotics when taking antibiotics for non-bacterial infections, not taking enough dosage, or not completing the full course of treatment. In other words, stopping taking antibiotics once you begin to feel better or once symptoms have disappeared. Antibiotics are also infused in the feed given to livestock, like chickens, bovines, and swine. This can generate antibiotic resistance in bacteria that can find its way into the food supply, threatening food security. Antibiotic resistance is also observed commonly in hospitals where antibiotic use is high. In case this is not frightening enough, some strains of bacteria have multiple resistance to antibiotics. Multi-drug resistant organisms are bacteria that have become resistant to certain antibiotics, and these antibiotics can no longer be used to control or kill the bacteria. What can you do in order to limit the risk of antibiotic resistance in bacteria? Wash your hands with normal, non-antibacterial soap and warm water. Wear protective clothing, like face masks and gloves, when interacting with infected individuals. Use antibiotics only when needed, and don't share your prescription with other people. Quarantine infected individuals to limit the spread of bacteria. Clean and disinfect care items, equipment, and surfaces daily, and get vaccinations ahead of times of high rates of infection, like before flu season. Natural selection is a theory. How much evidence is required to support a theory, and what sort of counter-evidence is required to refute it?